So, welcome back from coffee break to this particular session. I'd like to thank Bad and his team, actually, for the wonderful conference, this wonderful Clinim conference that you've all worked so hard at putting on. And also thank them for the opportunity for me to organize and chair this session. I'm Terry Allen. I'm a professor emerita from the University of Alberta, so I've retired and closed my lab, and I'm, well, semi-retired, I would say. And I'm a visiting professor at the University of British Columbia. So let me ask you, by a show of hands, who's heard of Pieter Cullis? Yeah, Peter, you'll be happy to know that there's a sea of hands that just went up, and that's the reason why I don't need to introduce you, because everyone knows who you are right now. I've known Peter for 44 years, actually, and so he's been working in the field of nanomedicines for close to a half a century. Obviously starting when he was only in kindergarten. And, he's done really seminal work starting out in kindergarten with the structure and function of lipids, and then working through developing various strategies to use lipids for clinical applications, ending up with some beautiful clinical applications that he'll describe to you in his talk. And all at the same time while founding over 10 companies, maybe 11 or 12 by now, Peter, or maybe, I don't know, every week it seems to be another one, but a large number of companies, and that's wonderful because it creates employment for scientists and economies, economic benefits and medical benefits, and so that his business he's not only, in other words, a very successful scientist, but also has been a great entrepreneur in the field of nanomedicines. So without further ado, Peter, take it away.
Okay, thanks very much, Terry. Can you hear me okay?
I'm assuming the others can.
Okay, so today I'm just going to, first of all, I'm very sorry I'm not there. I arrived in London on my way to Basel and was feeling rather under the weather, and tested for COVID, and I had it, and then I felt more under the weather, and I really didn't feel up to the coming there directly and possibly infecting a whole bunch of people anyway. So it's rather ironic that I would come down with COVID, but I seem to be recovering now. So I'm just going to talk in a very quick 15 minutes about the how it was we came to play a role in the design of the production of the Pfizer BioNTech COVID-19 mRNA vaccines, and and really give a bit of an overview of the 50 years of experience that led to this 50 years of lipids. I have a number of conflicts of interest, as Terry indicated. So you've probably heard that the mRNA vaccine is coming inside tiny bubbles of fat. This is both in the popular press as well as in the scientific press. This is really, I think, in our field, a little bit of a, it could be a slightly more sophisticated term than slightly bubbles of fat. There's certainly much more than that. It's really taken lipid nanoparticles 50 years to become an overnight success. And I'm going to go back to 73 basic studies on membrane lipids. And then in 1985, when we started developing these systems to deliver cancer drugs, and then in 1995 to deliver nucleic acid-based drugs. Now, I've spent most of this time considering two basic problems concerning membrane lipids. And this led to the mRNA vaccines. Now, you wouldn't think this is the precursor, but that quite directly to the nanoparticles that enable the vaccines. So, from 1973 onwards, I was very concerned with lipid polymorphism. That many of the lipids in biological membranes, about 50% of them actually, if you isolate them and put them into water, they won't drop to bilayer. They'll adopt these hexagonal phases. So, why are they there? And then the other topic is that of lipid asymmetry, that biological membranes, the lipids in biological membranes are asymmetrically distributed, just different distributions, different lipid species on one side of the membrane to the other. And so we started to ask the question, can we generate that asymmetry, and what are the consequences of lipid asymmetry? So very basic sorts of studies. Now, our studies on polymorphism really led to an understanding of membrane fusion, in the sense that the presence of non-bilayer lipids drives fusion of, say, unilamellar systems to form larger fused systems. And it's certainly difficult to understand how fusion can occur without a departure from bilayer structure, which is presumably provided by the ability of these non-bilayer lipids to adopt alternative structures. Our studies on lipid asymmetry led us to ionizable cationic lipids. In order to generate lipid asymmetry, we said, okay, well, let's put, say, 5% of a small percentage of an ionizable cationic lipid that she synthesized called DODAP. And this lipid has a property that can exist in the neutral form or in the protonated positively charged form. The neutral form is highly membrane permeable. And so if you have a lipid bilayer or a liposome with a low pH on the inside and a pH 7 on the outside, the neutral form can readily permeate across the membrane, gets protonated, positively charged, can't get back out. And so you generate lipid asymmetry. So that was what our basic studies gave us, and it gave us really the insight and the tools to be able to take these systems, or take systems that might have utility for developing, for delivery of nucleic acid polymers. Now, in '95, we started to work on developing LNPs for these nucleic acid-based systems. And of course, we required positively charged lipids. And you can see intuitively here, negatively charged RNA or DNA, positively charged lipids. You can see intuitively that we would, excuse me, that we would be able to generate, could be able to generate something where we had a lipid nanoparticle structure. Now, unfortunately, or fortunately, there's no positively charged lipids in nature because they're too toxic. And they are toxic, they're very toxic agents. And so we said, okay, well, how can we possibly encapsulate efficiently the nucleic acids without using these toxic molecules? And this is where we went back to to DODAP. This is Sean Semple, who pioneered this work. We used DODAP, the ionizable cationic lipid, and said, okay, well, maybe at the low pH where this lipid is protonated, we could associate nucleic acids. In that date, we were working with antisense, associate the oligos with the lipid. And perhaps if we had the right combination of lipids, when we brought it back up to neutral pH, we would be able to retain those contents. This turned out to be the case, and it evolved into a process whereby we just mix the oligo in water at pH 4, buffer at pH 4, the lipids in ethanol, say 50% or so of the ionizable lipid, together with PEG lipids, other structural lipids, phosphatidylcholine, and cholesterol. Mix these things rapidly, and you can suggest that the first thing to fall out of solution is the oligonucleotide, which is what we were working with, as I mentioned, surrounded by the cationic lipid. If you do this fast enough, then the other lipids, such as the PEG lipids and the phosphatidylcholine, will fall out of solution and trap these entities in what we term the limit size or limit size vesicles. And so this provided a technique for encapsulating RNA, DNA, or actually any negatively charged entity, a nanoentity, in a system that's relatively non-toxic, hydrophobic core, ideally suited to encapsulation of negatively charged macromolecules as large as plasmids, etc. Encapsulation efficiencies of 100%, scalable and reproducible. So then we asked the question, and this is work we did with Alnylam Pharmaceuticals, can we use these systems to encapsulate siRNA and then silence the gene in the liver following an IV injection? So this is a pretty ambitious project with the objective of transfecting hepatocytes following an IV injection. Now, LNPs get into cells by endocytosis. And so our job with the lipid nanoparticle is to help these entities break out of the endosome and release the RNA or DNA encapsulated into the cytoplasm. So in a sense, they must fuse with the endosome to release the nucleic acid cargo into the cytoplasm. Then we also we went back and looked at what the effect of cationic lipids is on negatively charged lipids, such as you find in biological membranes. And if you have a negatively charged lipid, phosphatidylserine, phosphoinositol, phosphatidic acid, whatever, any of those lipids, you combine it with a positively charged lipid, and it adopts these non-bilateral structures. And so we came back to lipid polymorphism some 20 years later as a rationale for how it is we could design these ionizable cationic lipids to maximize membrane fusion with the endosomal membrane. And so that the proposed mechanism is we have our lipid nanoparticle, the neutral form of the ionizable cationic lipid, and goes into the endosome. The pH is lowered. These get converted to positively charged form, indicated in red here. They combine with the negatively charged lipids in the endosomal membrane, and the what calls fusion and release of the encapsulated nucleic acid. So this is a simple picture. We don't have direct evidence that this is actually the case, but it has been very, very productive to follow this sort of rationale. Now we assess the in vivo potency using a factor 7 model, siRNA that's against to silence factor 7, inject that IV assay for factor 7 in the blood. And if the factor 7 gets into the hepatocytes, this factor 7 can be detected in the blood. And over the 7-year collaboration with Alnylam, we went from systems that had the ability to silence factor 7 in the liver from 10 mg per kg, actually it was a bit higher than that, down to 5 micrograms without increasing the toxicity of the system. And so we now had a therapeutic index greater than 1,000. We give 1,000 times higher dose, and we needed to see a biological effect before we saw toxicity. And I should emphasize that this is pretty important if you ever want to develop a drug, that your window in mice has to be way, way bigger, at least a factor of 100 before you would even contemplate going into the clinic. You always lose potency as you as you move up the animal kingdom. And so what disease should we treat? The conditions that Alnylam chose, HATTR, hereditary amyloid transthyretin induced amyloidosis, caused by transthyretin produced in the liver that has a mutation and forms fibrils, mainly in deposits all over, but mainly in cardiac and nervous tissue, usually fatal within 5 years of diagnosis, and a pretty horrendous type of progression, as you can see here. So this is where the joys of gene therapy become apparent. And what we have to do here is say, okay, well, this is the protein that's causing the problem. Let's shut it down using siRNA against transthyretin, and then reduce the levels of circulating transthyretin, prevent the production of these fibrils, potentially even getting stabilization and recovery if we can start to dissolve already established plaques. So a simple solution to a really a terrible disorder. So a phase 3 study was completed between 2016-17 over 16 months. With 148 taking the drug, or 77 getting a placebo, a sterile saline. The primary endpoint was a neural impairment score, and various secondary endpoints. had to be the most successful clinical trial I've ever seen. Certainly a profound improvement or a profound effect on the neural impairment score. If anything, these people were getting better. P-value that's, I don't know, I even bother to calculate this. And then the quality of self-reported quality of life, muscle strength, the body mass, et cetera, et cetera. So a stabilizing and possibly curative therapy for a previously fatal disease. This was approved by the FDA in 2018, the first FDA approval of an RNAi-based drug, certainly clinical validation of the lipid nanoparticle delivery system. So it really demonstrates the power of these gene therapy approaches. And so we went on at Cullis to then say, okay, well, if we can deliver sRNA, maybe we can deliver mRNA. And so this is work that was done subsequent to the Pieter going into the clinic. And so we wanted to find this out. And we can assay, this is a different assay now using luciferase model, injecting LNPs containing luciferase mRNA. Injecting that IV, and then following, as you can see here, some profound transfection in the liver, as visualized in this case by IVUS imaging. Now, what we found was that we can never make any protein we want by injecting LNPs containing mRNA coding for that protein, and this is just an example for erythropoietin. So seems like the liver does not mind making a few extra. It already produces 70% of the proteins in your body. It doesn't seem to mind making a few more. And so this obviously has huge therapeutic implications, and we were pursuing those in Cullis. However, we then were approached by Drew Weissman at the University of Pennsylvania, who'd been working with Katie Carrico to develop mRNA therapeutics as vaccines. And he said, we have a delivery problem. How do we get mRNA coding for these viral proteins into the muscles and immune cells in vivo? So the first study that was done was on Zika virus, that published in 2017 as a vaccine. So here we have mRNA that's coding for the Zika virus pre-membrane envelope glycoprotein, and this gave remarkable results. Really in total protection against Zika virus. So this is using in this case, I think it's intradermal injection of the lipid nanoparticle containing the mRNA coding for the surface protein, challenging at 2 weeks or 20 weeks, and total protection. So this led to a partnership between Acuitas and BioNTech. And in turn, when the COVID-19 pandemic hit, to a program with Pfizer using the same lipid nanoparticle that we've been providing them for a flu vaccine program. But in this case, of course, for SARS-CoV-2, we have mRNA coding for the SARS-CoV-2 spike glycoprotein. And so this was events that happened in January and February of 2020. And you all know the results of this, 95% effective against COVID-19. This was announced in November of 2020. A really spectacular achievement. Efficacy consistent across all ethnic, age, gender, reasonably well tolerated. I think they actually manufactured up to 3 billion doses in 2021 and also in 2022. So we really spent 27 years developing LNP systems, and it's been quite a journey from encapsulation of nucleic acids to the Onpattro story, to the Comirnaty, the mRNA vaccine story. An amazing journey. But it's certainly not over yet. This is really, in my view, the third generation of pharmaceuticals, gene therapies for protein replacement. vaccines and gene editing therapeutics. There are now over 300 LNP mRNA medicines that are in development. We've had a huge impact here. We're going to see things like a universal flu vaccine. I don't have time to go through the details here, but this is an LNP containing mRNA coding for 4 separate proteins within the influenza virus. And we get protection against challenge from a variety of strains of influenza, treatment for cardiovascular disease. In this case, the base editing PCSK9, just a single injection. And as you can see here, this is like a vaccination against atherosclerosis, the major killer. Treatment for cancer, producing proteins in the liver that then this is a bispecific antibody to bring T cells and tumor cells together, and comparing that to the recombinant protein monoclonal antibody administered IV, you can see it's behaving just as well, if not better. So LNP mRNA technology is now going, in my view, going to enable gene therapies in a spectacular fashion, and will be the next generation of pharmaceuticals, with applications, as indicated, ranging from vaccines to chronic diseases to various inherited diseases. Now, this has been the work of literally hundreds of people. I want to pay particular tribute to Mick Hope, Tom Madden, who I've worked with for over 50 years, well, over 40 years now, as well as others at Acuitas, Steve Ansel, Ying Tam, Barb Mui, Paulo Lin, Sean Semple, and the contributions, of course, of Alan Island. There's a great team we worked with there. My own group in UBC Biochemistry, and of course, Drew Eisman, who really got us involved. I hope there's a bit of time for some questions, so thank you very much.
Thank you, Peter. Now, because Peter won't be able to join us up here for the discussion period after, I think we'll take a couple of minutes from that discussion period to have questions for Peter. So are there questions, a couple of short questions perhaps that one might ask of him while he's still online here? So there are 1 or 2 questions.
Thank you for the great talk. I was just wondering how you see the development in the future, like in the role of the helper lipids on a LNP development. So what can be made better in the future?
Of course, right now we're constrained a bit to intramuscular and to IV administration and having the liver. We can target the liver pretty well, but not other tissues. This I think is going to be changed soon. We will get systems that can circulate for a long time. Right now, they're all out of the circulation in 15 minutes or so they don't have the chance to transfect other tissues. So the generation of long circulating systems that can transfect more globally, I think, is going to be the next stage. Also, the direct injection into privileged organs such as the brain, the eye, the ear, etc. We're going to see increased application in those areas as well. But I don't think there's anything really substantial standing in the way of being able to get transfection of pretty much any tissue. Now, whether we can do that specifically or not is another question. And I think the first stage is to be able to transfect cells anywhere in the body. The second stage is to get it to be perhaps more specific. And that's, I think, is going to be done by varying the helper lipids.
Thank you very much, Peter. I think we'll have to end there. Thanks. And get well fast.
Oh, yes. Yeah, I'm feeling a bit better now, but man, I was not great yesterday.